Hello everybody, I'm still a little bit tired, so I took the day off and hopefully I'll be back tomorrow. But I wanted to do this lesson so you guys didn't miss out, so I'm going to do it all by video. So today we are looking, I just wanted to recap a couple things with the Grape Lab, which you guys did on all the way back on Wednesday. Okay, a lot of you did really well, but I wanted to go over how we knew if these solutions were hypertonic, hypotonic, or isotonic. And a lot of you got a little bit different results, which is okay, but I think some of you probably got your grapes a little confused. And so that's why it's really important when we do experiments, it's important to label everything correctly. So your data, let's say in sugar water, you had it weighed 8 grams ahead of time, okay? For simplicity's sake, we're just going to say that they all weighed 8 grams, even though I know that's not really true. Okay, so they all weighed 8 grams. How did we tell if it was in a hypertonic solution? How could we tell which one of these was a hypertonic solution? Well, if we think about the word hypertonic, remember, hypertonic means the cell shrinks. We weren't using cells, we were using grapes, right? So in order for it to shrink, it would lose water, okay? So the sugar water, you should have gotten the weight went down, okay? And if the weight went down, that would make sense because it would mean that in my really bad grape that I'm drawing here, the water was flowing out and that caused it to weigh less, okay? So the change in weight, I don't really care about that column. I just needed you to see that from the first day to the second day, it lost weight. So that means it probably lost water and it shrank. So for a lot of you, the sugar water should have been hypertonic, okay? The grape juice, you should have gotten something similar. Now, what are the chances you actually got the same exact measurement? Probably not very high, not very likely. But you might have gotten something like 8.2 or 7.8. I know your numbers were more around 15, okay? So if you got something roughly the same, we could call that isotonic because it didn't change a whole ton, okay? So the white grape juice was supposed to be isotonic. Now, don't freak out if you're like, oh, Ms. Frankston, I got that all of them were hypertonic. That's okay, all right? It's your experiment. It's your results. So I'm not going to mark you off because you got different results. I'm just telling you what you should have expected to get. And this would make sense because the white grape juice would have roughly the same concentration of stuff, of water and sugar inside of it as the grape. And then the water, you should have gotten something where it increased in size, okay? And that's because water was going into the grape, right? Wow, I really cannot draw grapes on here. And so we would call that hypotonic. Okay, so I just wanted to recap that because I know I wasn't here for that for a lot of you guys. All right. So today we're going to practice osmosis a little bit more. We're going to have a quiz though on Wednesday. 7 or 8 questions, again, just a quiz online. Here's what's going to be on that quiz. I'm going to flip through to the quiz here. Okay, I will email this to you. Actually, it's already been emailed to you. Check your email. I will email this list to you, okay, and to your parents. So, first of all, what is equilibrium? Think about it. Who can tell me what is equilibrium? Remember we said equilibrium has the word equal in it. It's when there's a balance between both sides, right? So both sides have roughly the same concentration. Maybe it's 80% sugar on both sides, or they have roughly the same amount of sugar molecules or salt molecules, okay? What is the difference between osmosis and diffusion? Okay, you need to know the difference. Again, these are all going to be like multiple choice questions, but these are the things that are going to be on the quiz on Wednesday, okay? So, osmosis. Who remembers what is osmosis transporting? Osmosis transports water. So, osmosis is with water. What about diffusion? Diffusion is everything else besides water. Could be sugar, salt, Kool-Aid, chloride, potassium, whatever you want. It just can't be water, okay, because water is special. What are the characteristics of passive transport? We wrote down 4 things about passive transport, okay? Anyone remember, it always goes from what concentration to what concentration. That's one of them. From what concentration to what concentration. Goes from high concentration to low concentration. So you need to know that from where there's a lot, high concentration, to where there's a little. Okay? Both of them, well, passive transport transports things. It does not use energy. Okay? So no energy is needed. That's important. And it sometimes uses a protein. It doesn't need to use a protein, but sometimes it uses a protein. And does anyone remember what size molecules we use passive transport with? If it doesn't use energy, what size do the molecules have to be? The molecules have to be small. Okay, we can't transport big molecules because they take lots of energy, and passive transport doesn't use energy. Okay, so remember, osmosis and diffusion, those are both examples or types of passive transport. Okay? All right, can you predict where water will move? So if I give you a little diagram, right, like with the beakers, and I tell you that inside is 80% water and outside's 20% water, can you tell me if water is going to move inside the cell or outside the cell? And then tell me what will happen to the size of that cell. Right, so if water is moving in, we know the cell is going to get bigger. If it's moving out, it's going to shrink. And then can you tell me if a solution is hypotonic, hypertonic, or isotonic? So if my water is moving into the cell, what's my solution going to be? Think about it. It's moving into the cell. What's going to happen to the size of the cell? It's going to get bigger. And if it's going to get bigger, what kind of solution is it? It's a hypotonic solution, because remember, the cell's getting big like a hippo. Hypotonic hippo, okay? Isotonic means it stays the same. Hypertonic causes the cell to shrink. So today what I want to do is recap a little bit of osmosis. You guys are getting really good at this. I just want to go back and make sure that we fully understand it. So on your desk or somewhere around your desk, there should be a dry erase sheet. Okay? If there is not, ask the sub for one. All right? If you need a marker, the markers should be out on the table. You know where to grab them. Okay? So osmosis worksheet. You need to know how to do this. So it is very important that you're doing this. Just because the sub is here does not mean you get to sit and slack off. Okay? Because you're going to be tested on this. I guarantee you these problems will be on your test, which I'm going to have next week. Okay? So what you need to do is you need to draw arrows. All right, showing the way that the water is going to move. Okay? So just draw arrows showing the way the water is going to move. And then what I want you to do is label each solution as hypertonic, hypotonic, or isotonic. So let me go back to my sheet here. We'll do the first one or two together. Okay? So it says we have 90% water, 10% sugar, 85% water, and 15% sugar. So look at the water concentrations. That's all we're worried about is the water. Okay, we're just saying where the water is going to move. Which way is the water going to move? If we have 90% in the cell and 85% outside. Remember, it always goes from high, from where there's a lot, to low. So my water is going to move outside the cell. Okay, so I'm going to draw an arrow. Now, if it's moving outside the cell, okay, what's going to happen to the size of my cell? It's going to get smaller. And if it gets smaller, what kind of solution do I have? I would have a hypertonic solution. Okay? So, go ahead, do the rest of these. Okay? Sub's going to pause this video, and then we'll come back and we'll go over the answers to these in a second. Okay? Alright, so let's go over the rest of these. You should have them done by now. Okay? Alright, so number 2 here. Again, look at the water. 40% water, 90%. Water's going to go where? Into the cell. Okay, if it's going in, it's make it bigger, so it's going to be hypotonic. Okay, over here we have 75 and 80. Water's going to go from the 80, the big one, into the cell. So again, we're going to have hypotonic. It's really impossible to draw on here. All right, over here, water, 75 and 45. Again, it's going to go in. Okay, so we've got hypo. Then we have 90 and 63, so we're going to get water going out. This is an example of hypertonic. Now, over here we have 50-50, okay? What I do not want to see is you leaving this blank. They have the same concentration of water. You don't leave it blank, because remember we said, just like in the snowball fight, the molecules don't ever stop moving, okay? They're always moving. So what I need you to draw is an arrow going in and out, okay? That shows that they're going in and out, and there's no net movement. There's no movement or big movement of a whole bunch of particles in or a whole bunch out, but there's some moving in and some moving out. So this would be isotonic, okay? And then over here we have 90 and 90, so those are the same again. So in and out, that would be isotonic. All right, next one we have 82-25, so it's going to go out. So that would be hyper. And then over here we have 90 and 80, so that's going to go out as well. So hypertonic, okay? All right, awesome. So you need to be able to do one of these on the quiz on Wednesday, and then you'll need to be able to do more of them for the test next week. So what I need you to do right now is erase this sheet. Erase it. Make sure that your marker gets in the bucket. I am tired of finding markers on the floor, okay? These markers are expensive, the dry erase markers. So look around your desk, make sure there aren't any on the floor, and put them in the bucket at your desk, okay? So the next thing I want to talk about is something similar to this, but instead of happening with a beaker and a cell, it happens in a tube, okay? You know scientists, they like to make really creative names or not so creative names. So this is actually called a U-tube. Why? Because it's shaped like a U, okay? Now they call this a U-tube, not like YouTube where you go watch all of your videos, but literally U-tube because it is shaped like a U, okay? So notice, on this side, we've got a lot more concentrated stuff, whatever it is, sugar, okay? And on this side, we have less of those dots, less stuff. In the middle, we have what's called a membrane, just like the cell membrane. It can let water through, but it can't let these big particles through because they're too big. So what we need to do is predict which way is the water going to move, okay? Now, if you're cheating and looking at this arrow already tells you which way it's going to move, but we're going to tell you why. I want to tell you why, because you're going to have to predict this on the test next week. I'm going to give you an example, and you're going to have to tell me which way is the water going to move, okay? So remember, we always want to get to equilibrium, an equal concentration. So what's going to happen is if we were to, let's think about this in terms of numbers, right? This side's more concentrated. So let's say it has 80% sugar, okay? If it has 80% sugar, I'm just going to write S. What does my water have to be? If it adds up to 100%, what does my water have to be? If 80% sugar, what is the water? 20%, right? 20% water. And again, I'm just going to abbreviate it W because I can barely write on this thing, okay? Now let's think about the other side. The other side's got less sugar. So let's say it's only 50% sugar. If it's 50% sugar, what does the water have to be? Well, the water has to be 52 because it adds up to 100, okay? So now, which side has more water? Here we have 50, here we have 20. Which side has more water? This side, right? So water always moves from high, from where there's a lot, to low. So where's my water going to move? My water's going to move to this side. And we see that, okay? In this picture, the water increases on this side. Now notice, the water always wants to make it balance out the concentration. On this picture on the left, notice, we have a lot more dots in a little space compared to over here. In this picture, though, they're about even. That's because they have the same amount of dots in a roughly the same amount of water, the same concentration. And that's equilibrium, okay?